Welcome to everybody. I've got a slightly sore throat, as you'll hear, but I'll do my best. Hmm. Um, you guys in Key Stage 3 did some questions on the cells and the lungs, I believe. That was the sort of general topics that we did. Um, and I suspect you have some questions on your homework. Can we do 4, 6, 9, booklet 1 for year 7? Yep. And 1 to 12. That sounds optimistic. Um, we'll see. Right, I will begin with booklet 1, since they are the only questions today. Um, your booklet will have been booklet 2, the person who has forgotten which booklet it is. Surely it says it on the front. Yeah. Um, okay. Yeah, there's 1, 2, and there's 3. Um, so, someone's asked for 1 to 12. Let me have a look at some of these. Um, okay, I think I'll just go through this. But for the year sevens, I think it's reasonable to do quite a lot of this. I'll start in a second. The diagrams show 2 plant cells. Yeah, I must have clear my throat again. The diagrams show 2 plant cells. In which part of a plant would these cells be found? Cell X and cell Y, right? Basically, what you are supposed to recognize in this question is that we have a cell here. It's not helpful without the colors, but there is a cell here with lots of chloroplasts. That's what these are. These are generally found in the leaf of a plant where cells have lots of chloroplasts because that's where all the light goes. Cell Y, now this shape you should hopefully recognize is the shape of a root hair cell. That's what a root hair cell looks like. So on the roots, there are these tiny hairs, and they're called root hairs. Are made of cells called root hair cells. I'll do. The name of part B is pointing at the outside of this cell, which means that it is a cell wall. We have cell membranes in the inner layer and then cell wall on the outer layer, only of plant cells, as you may recall, not of animal cells. Give the letter which labels the nucleus. I'm not going to do that one. What's the function of the nucleus? I'll also not do that one because I think you should be able to find the answer to that somewhere. How can you tell from the diagram that photosynthesis cannot take place in cell Y? Okay, again, not the easiest question, but it helps if I add some color to this, right? These things are chloroplasts. They are green. Their job is to absorb light. This cell does not have any of those. If it doesn't have any of those, then photosynthesis, which is a process that needs light, cannot take place. So something about cell Y does not have any chloroplasts should work as an answer to that question. Um, which process? If you have any other questions, by the way, please tell me from your review booklets. Yeah, 1E, booklet 3, I'll come to that. Thank you. Which process takes place in both cell X and cell Y? The process of respiration takes place in all living things. So it doesn't matter whether it's a plant or an animal or bacteria. If it is a living thing, it does this thing called respiration, which is how that cell releases energy. Um, I'll just keep going. In which part of a plant would you expect to find that's a leaf again? I'm not going to put the function of the nucleus or the chloroplast. I've kind of already said that, or the cell wall. 2 labeled parts that are not present in animal cells. Animal cells do not have a cell wall, and they do not have chloroplast. They also don't have a vacuole. So any of those 3 would be sufficient there. Um, Photosynthesis and respiration. Okay, I think what I'm going to do very quickly, just to make sure everybody understands this, is I'm going to write down the equation for respiration and write down the equation for photosynthesis. Respiration is glucose plus oxygen, arrow carbon dioxide plus water. And the whole purpose of respiration, which you don't see in the equation, is that it releases energy. It's a strange pen. Um, Photosynthesis is the respiration equation backwards. So it is carbon dioxide, sorry, my writing's really messy at the moment, plus water gives glucose plus oxygen. So it's all of that in reverse. The other thing to mention is sometimes they put this on the equation. is that this cannot happen without light. So you need light to make it begin. And I think now that I've done that, we should be able to answer all of these questions. Carbon dioxide is produced. Produced means on the other side of this arrow, that's only respiration. Light is needed, that's only photosynthesis. It occurs in plants and animals. So respiration occurs in both. Photosynthesis is only in plants. So in plants and animals, that should be respiration. Oxygen is produced, again, produced is the other side of the arrow. That is photosynthesis. So if you know the equations, that becomes an easy question. Okay, question three. Again, I'll just tell you that as a chloroplast, because it's not always easy from these diagrams, because they're black and white. That is a chloroplast. You can figure out the function. The name of part E, I'm not going to tell you. You can figure that out. Two parts are present in plant cells, but not in animal cells. Ooh. I would say A, because A is the chloroplast, and B, which is the vacuole. I think you can say D as well, because it looks like it's pointing at the cell wall. How can you tell this is from a leaf and not from a root? From that question earlier, we saw that root cells don't have any chloroplasts. They don't have any chloroplasts because they're underground, and underground there's no light, so there's no benefit of having a chloroplast that absorbs light. Um, and so how can you tell? Well, it has chloroplasts. This shows the lungs and the trachea. The airway leads to the lungs. One of the lungs is drawn in section. In the walls of the trachea, there are pieces of stiff material called cartilage. Why is it necessary in the wall of the trachea? Okay, so um, these stiff pieces of cartilage are like strong material that is very hard to squeeze, right? If you try to press it, it kind of is in the way. It's too hard to press, which is quite important for your windpipe. Because you want your windpipe where you're breathing from to stay open and not have it squeezed in any way. So this stiff material is helpful because it helps to keep the trachea open. There are other things you could say, but that would be the most obvious answer, I would say. Um, question two, booklet three. Yep, I'm going to get to that. So I'll just get through this quickly. Sorry, I'm doing this quickly first. Shows one alveolus and its blood supply. Gas A enters the blood from the alveolus, right? So this is the lung, okay? And this is your blood. You breathe in oxygen for it to go into your blood so that it can be carried around the body, which means gas A must be oxygen. And we breathe out carbon dioxide. So carbon dioxide goes sort of into your lungs and then back out through your mouth. So that would be carbon dioxide. Give one reason why it's easy for gases to pass through the walls of an alveolus. So these things here, these they're called alveoli in plural, um, have very thin walls. By being a very thin wall, it means that stuff can get across it very, very easily. So that's all I would put there. I'd put thin walls. This shows a ciliated lung from the lining of the airway. What is the function of this cell in the airway? So cilia, um, essentially is like a bit like I had a sore throat just now. Um, you when you breathe in, you have this stuff called phlegm in your throat, which tries to catch any stuff that you breathe in that you don't want to go any further. But then what that cilia does, so if this is your throat, right, there's these little cilia hairs on the sides, and then you've got this disgusting stuff in your throat called phlegm, or mucus, and anything that you breathe in that shouldn't go any further gets trapped in here. But then we want to get rid of this somehow. So what these little hairs do is sort of waft it back up in that direction, and then you can spit it out or swallow it. And that helps to do that. So what it does, these cilia help to move mucus up. That's all I want to say for that. No, there's loads of questions here. Please explain one on booklet three. Yeah, booklet three. Don't worry, booklet three. I'm coming to it. Don't worry. This cell is affected by substances in cigarette smoke. What effect does cigarette smoke have on the cilia? I think if you let's say if it stops it working would be okay, or destroys them. Something like that would be okay. Okay. Give the name of the substance which causes addiction. It's called nicotine. Okay, so that is question four. Let me see what other questions. I might come back to this booklet later. 'Cause I'm not going to do every single question. Four, six, and nine. I'm just getting done. Um, a person has an asthma attack, find it difficult to breathe. An inhaler helps the person breathe more easily. Um, okay, during an asthma attack, it's closed and then it's opened again. Describe the way the airway changes when the inhaler is used and how this change makes it easier to breathe. So look, this is your tubes in your lung, nice and big and wide. But then during an asthma attack, look at it, it's tiny. That means you wouldn't be able to breathe in oxygen. But after using an inhaler, it helps to open it back up again. And so we want to say it opens um, or makes it bigger, I suppose. Yeah, makes the airway bigger. Surely you can say that opens it up. Yes, opens up is fine, which means if it's bigger, more oxygen or more air can get in. Yeah, so more oxygen can enter the lungs. That makes it easier to breathe. Okay. Um, the wall of the tube contains a ring of muscle. During heart attack, the muscle contracts and the airway can narrow. How does using the inhaler affect this muscle? It will relax the muscle. Because the opposite of contracting a muscle is relaxing a muscle. That'll be the exact opposite. Um, right, there seems to be quite a lot of booklet E. I've got three questions coming in. I will just do the last one from this one that has been directly asked. Nine. Right, the diagram is part of a blackberry plant. Photosynthesis, right, we should know the equation here. I wrote it down earlier. It is glucose. Um, we studied a blackberry plant growing in a shady place and a blackberry plant growing in a sunny place. We found that the plant in the shady place had larger leaves. Why is it an advantage for plants in the shade to have leaves with a large surface area? If a plant grows in the shade, then its problem is that it doesn't have much light. So by having bigger leaves, it's able to absorb more light, it's able to catch more sunlight, right? So it's an adaptation to try and help it stay alive and get more light by having bigger leaves. Which part of the cell makes the leaf green? You can say chlorophyll or chloroplast. That shows a similar question from earlier. I'll let you do that one. And yeah, that's all for question nine. Right, I'm going to move to booklet three. There's quite a lot of questions from there. I might come back to this one. I know somebody asked for all of it, basically, which is unlikely. Um, right, let me just do all of question one, and then we'll see what happens. What is the function of the nucleus? Okay, so that controls the cell. Which type of cell? Name one type of cell that does not contain a nucleus. The best answer is probably a red blood cell. Red blood cells don't have a nucleus because they carry oxygen and there's more space to carry that oxygen. Simple diagram of the cell in figure one. Oh, geez, really? It's an animal cell. So just do something like that. Label the cell membrane, label the nucleus, label cytoplasm if you like, label some mitochondria and ribosomes if you like, but I think that's easy enough to do. Name one structure found in a plant cell. I won't do that. Right, this one is tricky. So in fact, it's basically impossible actually to get right without my assistance. And I'll tell you why. It is because it requires you to get a ruler out and measure something. But the problem with measuring this is that um, because we've printed it on like smaller paper and made it bigger and smaller, the actual measurement is not going to be accurate when you measure it on your page. But I will tell you what you're supposed to do. Hmm. So it tells you the real length. So what we have done, we have zoomed in on some cells with a magnifying glass, not magnifying glass, microscope. With a microscope, we have zoomed in. And we are told that the real size in real life, because it's so small, is 0.06 millimeters for this. Oh, but 0.06 millimeters is like, I mean, I can hardly make it with my between my fingers. It's very small. It is not the size of what you're looking at right now because that's been magnified. So when you see this equation about magnification, the real size is what it sounds like. It is the real size in real life. The image is what you see when you look down the microscope. It's been made bigger. So what you're supposed to do now is look at this and using a ruler, measure this to here. But the problem is, as I said, you will almost certainly get the wrong answer because whatever you're measuring probably isn't the correct size because we've zoomed in and stuff. But this distance, I can tell you, if you did correctly do this, is supposed to be 24 millimeters, so 2.4 centimeters. I don't know if it looks like that on your page, but it is supposed to be 24 millimeters. What you do with that 24 millimeters, and I've said millimeters there because this is also in millimeters, it's important to keep units the same when you do formulas. Put that 24 there, and using a calculator probably, do 24 divided by 0.06, and you will find that the answer turns out to be 400, which means that we have zoomed in using the microscope 400 times onto this cell to be able to make it this big as you look at it. Um, right, then you might not know this, but an electron microscope is a microscope that has a higher magnification. It means it can zoom in further, and a higher resolution, which means that it gives you a clearer image. Even when you zoom in, you may know that when you zoom into things with a phone, for example, it's not very clear. It gets all blurry. But the resolution being higher means that it doesn't get blurry either. Very powerful. Um, right, that's 1E and F done. I'll have a look at 2A and 3A. Cells containing a solution of salts and sugar. Cells. A student is investigating how cells change when they are put into water. The student looked at a plant cell using a microscope, adds water to the cell. The plant swells up. Explain why. Well, this is something called osmosis. If I have a cell and I surround it with water, the water moves. Let's say this is all water, a very high concentration of water, so lots of water. Um, there is something called osmosis, where water moves from a high concentration to a low concentration, which means that the cell gets swelled up like a balloon. Yes, this will be on our YouTube afterwards. Um, it'll take a while. It might be tomorrow that you get to watch it. But explain as fully as you can why the plant swells up. So we want to say because water enters the cell or goes inside the cell, you want to mention osmosis because osmosis is when water moves from a high to a low concentration. Is it only 2 marks? No. And also the hard mark here is the last bit, which is the cell has a cell membrane. Cell membranes allow things to go through, some things, and we call them partially permeable. That means some things can get through, some things can't, but water can. That is how you answer the question. So that basically is the definition of osmosis, that is when water moves from a high to a low concentration um, through a partially permeable membrane. 1E, 1F, 2A, 3A. Red pigment found in red blood cells. It's called hemoglobin. Hemoglobin is this stuff that's found in red blood cells, and its job, which is the next question, is it can sort of lock onto the oxygen, but we want to say um, its job, what the hemoglobin can do is that it can carry oxygen um, from the lungs, because that's where we breathe it in, to your tissues. You can say cells or tissues here. It doesn't matter. Quite hard to get the second mark there. One other way in which the structure of red blood is different from the structure of a white blood cell. The key thing to try and remember, if you remember nothing else, is that red blood cells do not have a nucleus. Okay. Sorry, I'll write that clear. There you go. Carries oxygen from the lungs to tissue cells. Describe another way in which the structure of a red blood cell is different. Red blood cells do not have a nucleus. That is one thing to try and remember about red blood cells. They are basically the only cell in your body that doesn't have a nucleus. 7D from which booklet? If you are going to say 7D, if you could tell me. Um, A6, part 1 and 2. What is the relationship between the maximum rate of... Okay, right, let's read this. I think people don't do very well on these questions. 7D from which booklet? Okay. Oh, yeah, now. Yeah. Okay. In an investigation, four groups of athletes were studied. The maximum rate of oxygen consumption for each athlete was measured. That basically means the most oxygen they're able to breathe in. That's what we mean by that. Um, the maximum rate of oxygen for each athlete was measured, and the mean for each group was calculated. If you need help from booklet 2, then you need to say which question number you need help with. I don't just go on to it without specific question numbers. Um, the athletes then ran 10-mile races, and the mean of the best time was calculated for each group. Okay, so Group A, on average, they have got this much oxygen per minute or whatever, whereas you can see it's going down there. But let's have a look at their times in a race. You see how that's the fastest time? A low time is a fast time. So it seems as though if you're able to consume more oxygen, you can do the race quicker. Whereas if you consume less oxygen, it takes you a longer time to do the race, which kind of makes sense. The more oxygen you can get into your body, the more energy you can release from respiration, and that means you should be able to run faster. That's why smoking is bad, for example, because it results in you getting less oxygen into your body all the time, and so you run out of breath quicker. And so if you have to explain the relationship here, this should be quite easy. The relationship suggests that as the maximum rate of oxygen consumption increases, the time gets faster. Or there's several ways to say the same thing. Or let me say it in a different way. The higher the rate of oxygen consumption. See, this is getting higher as we go this way. But what's happening to the time? It's getting lower. There you go. That's it. Suggest an explanation. So I think I kind of sort of already explained that. Whenever you have a question about, this is very common, by the way, it's really good to get good at this now. Any situation where somebody is getting more oxygen, okay, or faster. So the faster the oxygen enters the blood, so if you can get oxygen into your blood quickly, then the faster respiration will take place because respiration is a process that needs oxygen. More respiration leads to more energy because respiration is all about releasing energy. And if more energy is released, you can run faster. And so your time for a 10-mile race decreases. So just so you understand, again, I think people often are not very strong at these sort of questions when there's numbers involved and data. But you should recognize that this group has got the sort of gets in the most oxygen. That's what this is, maximum rate of oxygen consumption, is how quickly they can get oxygen into their body. And if they can do it quicker, so more can get in, they're able to run faster. And you have to explain why, because more oxygen leads to more respiration, that leads to more energy being released. Respiration, what is the purpose of it? I'm not doing that question, am I? Surely not. 7D part 2. So, what is the name of the process of respiration when oxygen is not available? Firstly, that is anaerobic respiration. Anaerobic. Anaerobic respiration basically means without oxygen. But then what does that do? So, this tends to happen during, um, well, in fact, there's an equation for anaerobic respiration in animals, which is glucose, arrow, lactic acid. This is like when you have a stitch. So you get this when you um, do like intense exercise and you're not able to get oxygen into your body quickly enough. And so your body has to resort to this process, which helps to release a small amount of energy But the lactic acid it produces is painful. That's why like a stitch is painful, or if you do lots of exercise, it starts to hurt after a while. That should be enough for you to get that. And eight. Ooh, A. Um, as they go higher up a mountain, mountaineers take less oxygen into their bodies with each breath. So this is now almost the opposite of what we were talking about earlier. So before we were talking about how getting more oxygen is an advantage. Now it's saying that these mountaineers are going up somewhere and there's less oxygen there. So if there's less oxygen, they're going to have less energy because oxygen is needed for respiration to release energy. This is shown in the table below. So at the bottom of the mountain, it tells you how much oxygen is taken into the lungs with normal breath, that much, and how much oxygen goes into the blood. But then if you get to the top of the mountain, look at these numbers, they halve. So there's half as much oxygen getting into your lungs and half as much oxygen getting into your um, blood. At the top of the mountain, they only take half as much oxygen into their lungs. That's what we said. How does this affect the amount of oxygen that gets into blood with each breath? Well, that also halves, doesn't it? You see that there? So that's half, but that is also half. Okay. And that would actually be 2 marks. Why do the cells need oxygen? Well, the reason you need oxygen is for respiration to release energy. I do not ever want to see anybody answering a question about why do you need oxygen with to breathe, because that doesn't make sense and it doesn't tell you anything. Breathing is the process of getting oxygen in. It doesn't tell you why you need it. It's because we need to release energy using this oxygen. Um, okay. So releases some energy, but lactic acid is painful. Anaerobic, right? That would be fine. Half as much. Let's write that again. Okay, half as much. Yep, fine. Um, again, if you need any help from any of the other booklets, then you needed to have asked the question number earlier. I'm not going to do anything from booklet 2 now because you need to ask and not just say, when's booklet 2? I need question numbers if you need help with a specific question, assuming you have tried it all by now, surely. That is all of booklet 3. Well, leave the question anybody asked me. Does anybody have any other specific questions from any of the 3 booklets? 2A booklet 3, yeah, sorry, I'll do that again. I'll show you. Um, okay, I'll read that. I'll write that again. 2A. So, the idea of this was that the reason a plant cell swells up when you put it into water is because water enters the cell by osmosis. And then whenever you mention osmosis, try to mention this partially permeable membrane. It's quite hard for you to remember this right now, but if I say it enough, you'll remember it. Permeable. Okay. Um, to a part 2, booklet 2. To a part 2, booklet 2. Um, yeah, I don't think I should do that. So, how can you tell it's an animal cell and not a plant cell? Remember, plant cells have extra features like chloroplasts and cell walls. Um, so that should just about do. Anything else? Yes, and vacuole, very good. Correct. Indeed, indeed. Yeah. I'll give you a few minutes if you want, if anybody has any other questions from their booklet, specific questions if possible. Um, 2B feels like you're doing this at the moment, doesn't it? 2B. Um, Oxygen will diffuse into the cell. Why? Okay, diffusion. I'm not going to answer this question directly. I'm going to tell you that diffusion has a definition. It is the movement of particles. Sorry, there's something wrong with my pen. Um, from a high concentration to a low concentration. Now, you should be able to look at this picture and see that there is lots of oxygen outside this cell. In other words, there is a high concentration of oxygen outside the cell, and there is a lower concentration inside the cell. And so diffusion is when these particles move from a high to a low concentration. Um, um, I believe you. 2B booklet 3. I thought we'd done that. Oh, when animal cells are put in water, they swell up and then burst. When plant cells are put in water, they swell up but do not burst. I'm just going to tell you this. Plant cells have a cell wall which helps to protect them from bursting. I'm not going to write that down. As you know that plant cells have a cell wall, the extra cell wall gives them more support and strengthens them, which means they're less likely to burst as compared to animal cells. It's just having like an extra, I don't want to say an extra door, more security. Anybody got anything else? Otherwise, I will stop now and then return in 10 minutes or so to do something to do with the nervous system and other stuff. Apparently, Arsenal just scored. Thank you for that. I need help. I don't want to pick geography or history. Um, my advice with that is when I was in you're in year nine, aren't you? It's almost exactly in your situation. I was in exactly your situation in the sense that I didn't know whether to pick geography or history, and my decision in the end was determined by which one I did better in, and that was it. I'm not saying that you should do that, but I'm saying that is a factor. Consider which one are you likely to do better in, or which one are you doing better in, which is often the subject you tend to like a little bit more, and so on. And that is a good indicator. Whichever one you do better in is often a okay. Okay, I mean, if I appreciate you're good in both, but all I can tell you is my story, which is weird that I still remember this. But at the end of year nine, I had a history exam in which I scored 90%. I had a geography exam in which I scored 93%. It's weird I still remember this from a very long time ago. And I picked geography purely because of that 3%. I'm not saying you should do that. I'm just saying that's what I did. Um, but just as, or if you're good in both, decide which one you think you like more. Can't you do both? Can't you do both? Are you not allowed to do both? Because I could have done both, but I decided not to. Okay, well, there's nothing else coming now. So I am going to pause for 10 minutes, and then I will return. If you do still have some questions, geography is boring and history is interesting. Well, do history then. Done. You've answered your own question. Sorted. Right. Okay. I'll be back in 10 minutes. If you do have any still Key Stage 3 questions, then I'll come back a couple of minutes before and get through a couple if before I start the GCSE stuff. Which cells adapt for receiving? Nerve cells, neuron. Yes, that's correct. Very good. Yeah, 10. Yes, let's go. 1E. Have a look. So, right. Describe how these structures help to coordinate a reflex action. So the answer to this question is basically always the same. Um, okay. Which question number are you looking at? Sorry, the one with this. I'll just answer that quickly and I'll go on to this. The one with the nerve cell question that you're asking me. Where is it? Oh, that's because it's a. Oh, it's because it's D. It doesn't say it because that's the shape of it. So it's D out of all of those. Right. Um, 1E. So this answer is always basically the same in that you need to say exactly what happens in this process. So what you should have learned, hopefully, is that the receptors are the beginning of this process. That's what detect a change in your surroundings, which is called a stimulus. Receptors detect a stimulus. They then send information as electrical impulses down. This neuron A is called the sensory neuron. That's the beginning of the journey. Then the sensory neuron here connects to the relay neuron. Sensory neuron, I'll write it again. Connects to the relay neuron. And then you can say that the connection between two neurons is a synapse. This then connects to the next neuron, which is called the motor neuron. To this thing called the effector. And the effector then carries out the response, which could be like moving your hand away from a hot object. So, for example, this it could be where you detect a very hot object, and then all of this happens, and then your arm moves away from the object really quickly. And that is basically it. So when you answer these questions on reflexes and so on, you want to mention these words that are highlighted. Really, those ones. You can mention this synapse as well if you like, but these are kind of the important key words that you want to mention. Question 12 on the 10 quadratic equation book. Yes, I'll do that just after I do this. I appreciate that you're behind in terms of the um what's it called? Yeah, the questions, the booklets that everybody else is doing because of the fact that you attend on a Thursday. Right, I'll get through these 3B, 4B, 6B. 3. Okay. Explain why a man with a damaged spinal cord cannot feel a pin. So this is almost the same as the previous question, really. Except you just need to understand that if you have a damaged spinal cord, that middle bit doesn't work. So you'd say that the receptors in your toe, in this case, detect the pain. But because your spinal cord is damaged, the information cannot pass through the nerve cells. and it goes to the brain. So it cannot, that information can't get to the brain, and brain is where you feel the pain. So you're not going to feel any pain because the information that needs to get to the brain can't get there. Something like that should be enough. Um, 4B and 6B. 4B, describe how information travels from light receptors in the retina to the brain. So again, it's basically the same thing, which is that the electrical impulses go from your receptors in your eye, um, go down through the sensory neuron. That's the first one. I'm just going to put SN for sensory neuron. Um, how many is this? 3 marks. Electrical impulses go through the sensory neuron. And then there is 1 mark here for mentioning the optic nerve, which is quite difficult to mention here, but that I'll leave it like that. Because they're the main important bits to try and remember. So that's what happens. Your light shines into your eye, it goes down, the impulses go through the sensory neuron to your central nervous system, and then you can respond to the bright light or whatever it might be. Um, and 6B. Right, 6B. Right. Reflex actions involve nerves. Hormones is a chemical response. Now, hormonal responses travel in the blood, and so they tend to be much slower. So you want to say that a reflex action is much faster. Um, and also, you want to say that there's a few different things. Reflexes tend to only affect one part of the body, whereas hormones affect the whole body. You could also say that reflexes last for a short period of time, whereas hormones last for a long period of time. All right, 7B. I don't know where 7B is. Um, has it been taken out? Let me just check something. Aha, right, I've got it. Sorry, it's a different booklet. 7B. Okay. So that's just one of the mouse, isn't it, at the end? I've got the wrong one open. To be honest, this is the answer to this question is almost exact. It is exactly the same to the answer to the first question that you asked me, which was the whole idea. I'm going to put the keywords in. How does the mouse respond to the stimulus? So you need to say that the receptors detect the stimulus. Then you want to mention that electrical impulses go down the sensory neuron. It's exactly the same thing. Then to the relay neuron. Then to the motor neuron. These are the order in which these things happen. Then to the effector, which will carry out a response. Though these are the words you need to say when it talks about any sort of reflex response, when anything responds to danger in some way. Um, Electromagnetism, we do it in Both. Oh, we do it in year 11. That's right. We do it in year 11. Specifically twice actually because it's quite complicated. So electromagnetism we do at the beginning of year 11. That's when we actually have it allocated. But as I say, if you need specific things on it, first of all, there is a video related to it, which I made, which you can watch, that you technically have access to, but you have to ask me if you don't have access to it. So you can read up on it or watch stuff on it as to what would happen in that lesson. In theory, remember, all of you do have access, but you just have to check if you know how to access it. All of the stuff that we teach across the GCSE years. So yes, that's the answer to your question specifically. Right, I'm just going to go back and do one of the questions from last time, which was a maths question. Um, in fact, I'll probably have it here. Um, yes, I do. Um, okay. Why don't I get rid of all of this and explain it? Okay, difficult question, so don't worry. Hmm. Right, here are two rectangles. Show that the shaded area is given, right? If you wanted to find the shaded area here, the way to do it, if you had numbers, so put it this way. If I said to you, this is 6, 3, and this is 2 by 1, and I said I want you to work out the shaded area here, the way you would do that is you'd work out the area of the big rectangle, which is 6 times 3, and then take away the smaller rectangle, which is 2 times 1. That's how you would do that. So we're going to do this in exactly the same way. There's just weird algebra everywhere. So this side is 3x, and this side is 4x plus 1. So 3x times 4x plus 1 is the area of this entire shape. From that, we're going to take away the smaller rectangle, which has an area of 2 times 6x minus 3. Okay, so that's the first thing to write down. Then you need to expand the brackets and simplify. 3x times 4x is 12x squared. 3x times 1 is 3x. Minus 2 times 6x is minus 12x. And minus, be careful about this, minus 2 times minus 3 is plus 6. Very easy to go wrong there. And if you simplify this bit, you get 12x squared. 3 take away 12 is minus 9 plus 6. And that's ended up with this. Then it says that that shaded area, which you worked out with x's, is 6. So I'm going to write down 12x squared minus 9x plus 6, which was our shaded area, equals 6. Then solving this is involves, why this color is here, subtracting the 6. So you get 12x squared minus 9x equals 0. Then taking out the x to get, well, what you could actually do now is divide by 3 as well to get 4x squared minus 3x equals 0 because they're both multiples of 3. Then take the x out and you get 4x minus 3 equals 0. You ignore that one because it doesn't do anything. This one equals 0 means that x is 3 over 4 if you add the 3 and divide by 4. Um, right. So I will then. I think that's it really for the questions that we specifically asked. And then yes, you may not have got to the brain and the well. What do we do? Oh yeah, 'cause it was a different thing you were doing, so you wouldn't have done that. But I can do some of it now. But it would be something that I will keep doing, the brain and the eye and all these extra bits. Um, I'll open up the textbook to do this. So in a topic called homeostasis, which is what everybody actually did in year 10 as well, the nervous system. It extends on to um, stuff about the brain and the eye in triple science. So I will just show you the bits you need for that now via the textbook. I think is the best way to do this. It takes quite a lot of remembering the eye at least, but the brain not so much. The brain's quite easy. Okay. Um, this is the brain. Right. The first thing to say about the brain, okay, it was a very common question you get about the brain is why is it difficult to study the brain? Put it that way, right? And you want to make sure you have in your mind the fact that the brain is a complex and delicate organ. They're the sort of words you want to say. And because of that, it is very difficult to sort of study the brain without affecting the person whose brain it is. Um, so that's why it's very difficult to study it. So we'll come to that in a second. But you need to know that there are three main regions of the brain. There is the top bit, which is what you would probably think of if I said picture a brain, the sort of wrinkly bit at the top. It's called the cerebral cortex. And you need to know what it is responsible for. It is responsible for these kind of things, your sort of conscious memories, consciousness, your intelligence, your language that you speak, your memory is for those sort of the key important things, I suppose. Then you have something at the bottom, which is called the medulla, which is the opposite, I suppose, of the previous one. It controls unconscious activities, things that you don't have to think about, like your heartbeat and your breathing. It doesn't affect it's unconscious activities that are important. And then we have this cerebellum at the back, which is all to do with your movement, your muscle coordination, and that, which is why if that bit gets damaged, it can often affect your ability to move. So on. And I mean, they're the main three bits to it. And then this goes back to how we study the brain. So as I said, it is difficult to study the brain because of the fact that it's very complex and delicate. It is very difficult to study it without causing damage and stuff like that with the brain. But this is the way that we do it. We have information with regards to patients that have brain damage. So if somebody has brain damage and that one part of their brain has been damaged, and it has some other effect on their body, then we can comfortably associate that part of the brain with that part of the body that's not working or that's affected. So that's kind of how we've learnt more about the brain. So an example here, if an area at the back of the brain was damaged by a stroke, the patient went blind, then you know that that part of the brain at the back has got something to do with their eyesight. You see what I mean? That's how we sort of managed to learn a little bit more about the brain, unfortunately, from people who already have some sort of damage to their brain, and then we can see what effect it has on them. We can, and this is a bit of a risky thing to do, but we can stimulate the brain by pushing a tiny electrode into the tissue and giving a small zap of electricity. And then we see what happens. So if somebody zapped a part of your brain and then, I don't know, your arm shot up, then you know that that part of the brain is associated with that muscle. So, well, that's exactly what this says, actually. When a certain part of the brain, known as the motor area, is stimulated, it causes muscle contraction and movement. So we are able to do that. And then the third way in which we have learnt more about the brain is by using MRI scans, which produce very detailed pictures of the brain's structures. And we can, for example, if someone's listening to music, you can see which part of the brain is working at the moment. And then we can link those two things together in a way. So that's kind of the bit about studying the brain. And there's not much more to the brain, actually. It is knowing the three different parts of it, what each part does, and understanding how we study the brain and why it is difficult to study the brain. Essentially, that's all there is on the syllabus, really, to do with it. Okay, then the eye is more complicated. So I'm not going to go through every single label here, but there are some labels of the eyes. But I suppose the bit about this that is probably worth a bigger question is you can get asked about how this lens can change shape and why it changes shape, and why this pupil changes shape and how it changes shape. So think about two different things, why and how those two things happen. So let's start with the pupil. The pupil is like the central black part in your eye that can get bigger or smaller depending on how much light there is. It's actually related to reflexes. If you have a question from the year 10 thing, then just ask me. But I have done some of them. If I've already done one, then I won't do it again. But I have done all the big ones, and you can watch it back on the video. I think the big ones were generally asked. So the pupil can get bigger or smaller, and it gets bigger or smaller to allow more or less light in. It is an example of a reflex, actually, just going back to the things I was doing before. A reflex is when you have a fast automatic response to something. So if someone shines a bright light into your eye, your pupil automatically gets smaller. And similarly, when it's dark, it automatically gets bigger because it's trying to let more light in. How does it do that? Well, this is the bit where you have to remember this bit. Okay, so if there's one thing you need to remember is what happens when it's very bright to make the pupil smaller. There are two sort of interacting muscles here, a bit like your bicep and your tricep. When one relaxes, the other contracts. In the same sort of way, there's these circular muscles and these radial muscles, and they do the exact opposite thing to each other. When one contracts, the other relaxes. So when it's really bright, we want your pupil to get smaller, right? So I don't know how I'm doing that. Smaller, right? Um, and it does that by these circular muscles contracting. These are the words you have to remember. And the radial muscles relaxing. Okay, so to make the pupil smaller in bright light, circular muscles contract, radial muscles relax. And then the exact opposite happens when it's dark. You need to make your pupil bigger, so your circular muscles relax and your radial muscles contract, and that causes it to get bigger. So trying to remember which way round that is the tricky bit. Okay, so when it's bright, if you try to remember bright first, okay, so when it's bright, the circular muscles begin with C, contract, begin with C, and the radial muscles begin with R, relax, begin with R. And then the opposite happens when it's dark. Um, 6 and 7, I'll show you the answers. I think I've done those. And then lastly, for the eye, the other thing that you need to know is about the lens in the eye. In a similar way, there are these two muscles called ciliary muscles and suspensory ligaments. They control the shape of the lens in your eye. And the reason that needs to change shape is that it allows you to see things close up or really far away. And the lens has to change shape for when you're looking at things close up or when you're looking at things further away. So when you're looking at things close up, the lens has to be like short and fat, as shown in this picture. And it does so by this happening, the ciliary muscles contracting and the other ones, suspensory ligaments, relax. And that makes the lens go fatter. Whereas when you're looking at things further away, you want the lens to be sort of longer and thinner like this. And so the opposite happens. The ciliary muscles relax, suspensory ligaments pull tight or contract. So if you remember one of these, this is the thing, the way to do this is to remember one of them. Just go, okay, if I'm looking at something close up, what happens? And then everything opposite happens when you're looking at something far away. Or remembering what happens when you are in bright light. Just remember that much, and then you should be able to figure out that the opposite happens when you're in the dark. Um, and then there's a little bit about correcting vision defects, which is this idea that if you're long-sighted, it basically means that you can't see things very well that are close up to you, and it's because your lens is the wrong shape. It's not bending the light enough. The light should be focusing on the back of your eye like this, and so by wearing some glasses, we can fix that. Similarly, if you're short-sighted, it means things further away you can't see very well. That's because your lens is too fat, because it's not thinning, because your muscles aren't working properly. And so again, by wearing some lenses, you can bend the light the right amount so it focuses on the back of the eye. Sometimes you have to complete these diagrams. Other treatments, contact lenses, laser eye surgery and stuff, and that's pretty much it for the eye. The tricky thing really is not in this topic, it's in physics when you come to waves and you have to learn all these lenses and all these ray diagrams. That's when it gets complicated. Um, just going back to the year 10 homework, 6 and 7. Nope, not that one. So, question 6. I shouldn't need to mention these, but the structures involved here, I think that's going to be a receptor. Um, this is going to be like your central nervous system, this whole thing here that's labeled here. And then this is your effector. That's the one that responds to the action. Um, this is a bit harder, but how is a reflex action different from hormones? Hormones act more slowly. Reflexes are very fast. And reflexes generally only affect one part of your body. Like, let's say if I throw something at your head, your neck would move your head away. That's it. It's the only one part. Whereas a hormone like insulin affects the whole body. It goes all around your body and it affects the whole body. That's the two key main differences between those and those. Um, 3, I'll just see what 3B was quickly. 3B, I did answer earlier, but I'll just explain again. If you have a damaged spinal cord, the problem, it's quite a hard question, this actually, but the reason you feel pain is because if I don't know, bang my hand on the table right now, then the receptors in my skin are detecting that pain. They are sending impulses to my brain, and my brain is detecting that pain. If your spine is damaged or your spinal cord is damaged, those impulses are not going to reach the brain, as in they're going to travel from here to the central nervous system, but won't go up to the brain because the spinal cord is damaged, which means that I won't feel the pain because they have to reach the brain for me to feel the pain. So that's essentially the answer to that question. Um, and then there was also question 7. Was that the one that was on the other page? It was. I'm printing off the wrong ones of these. Um, Question seven. I assume it's seven part B, but three receptors which a mouse might use. I'm not. That's not on your screen. I'm just on the other screen. Three receptors a mouse might use to detect food under natural conditions. Light receptors to see things. Smell receptors to smell it, or and taste receptors to be able to taste it. I think that's probably the answer. Remember, your receptors. A lot of your receptors are just your five senses: sight, smell, sound, taste, and touch. I mean, that's essentially what they are. But there's other ones. There's temperature receptors, there's pain receptors, and so on. But in principle, that's what they are. So if I said to you, what senses do you use to detect food? Well, you need to be able to see it. You might want to be able to smell it. You might want to be able to taste it. So that's the answer to the question. And then part B of number seven is this one here. So this is very similar to there was an earlier question where you had to describe a reflex. It's the same thing. These key words are what you need whenever you are explaining a reflex action. You need to explain that there's receptors in your skin. You need to explain that they send electrical impulses. You want to say that's how we send information in our nervous system. Electrical impulses down the sensory neuron. That's the first neuron that goes to the central nervous system. If it's a reflex, then the sensory neuron connects to the relay neuron straight away, which connects to the motor neuron, which is what I've written down here. The motor neuron then sends the impulse to the effector, which might be your arm to move it away from a hot object, and that carries out the response. So these kind of words essentially answer any question involving reflexes or how you have a response in your nervous system. It's always the same sort of key words and steps. It's just applied to a mouse in this case. Otherwise, it is exactly the same. Um, Anything else? Coffee. Is there anything else I should tell you guys? Just as a reminder for those of you that I don't think there's very many people here in year 11, but if they are, then the it's getting closer now, but I know it's still quite far away. It's 2 months away, but from the beginning of May, um, as a reminder, I will be available every day for a short amount of time. Should you have any questions during and before your exam season of 6 weeks, every day I'll be here. Um, from about 7 o'clock. I think from about 7 o'clock, pretty much every day for about half an hour to an hour, um, to do anything really. Ramadan is going fine, thank you. All is well. You guys don't get to experience this that much because you come at a different time. But everybody who comes here during the hours of 5 to 7 gets some pizza. I've got loads of it today, so they all just feast on pizza during their lesson for 10 minutes. Um, right. It seems as though there's no other questions. But is there anything else that I could just do next week? I'm going to do something. I'm going to do something. I'm bored, so I'm going to do something. Next week, most of you, all of you, are going to be doing some chemistry. Um, I think the year 10s and stuff might be doing organic chemistry, might be. And the year 11s are doing the required practicals, the chemistry required practicals, that's what you're doing. But I'm going to do something completely different just for a moment, chemistry related. Um, I am going to suggest a question and let you think about it. Okay. Right. Imagine that was a question and you had to think about it. Quick question. What is a synapse? A synapse is a connection between two neurons. Let me quickly, whoa, do this. While you think about that question, have a think about how you would need to go about answering that. The how is more important than the anything else. Okay, just very quickly, the synapse. Here we go. That is a synapse. So we have a neuron. Anyway, um, there's for some reason, don't ask me why, there is a gap between from one neuron to the next. There is a gap there. That gap is called a synapse. Literally just that gap there. No, yeah, this is a synapse. You are meant to understand what happens here. So we have electrical impulses um, that get to here. Now, they'd have nowhere to go because there's a gap. But at the end of a neuron, there are some chemicals. And what happens is that these chemicals, when the electrical impulse hits them, the chemicals diffuse from a high to a low concentration across the gap. And when they go across the gap, they trigger another impulse, and so it continues. So it doesn't stop. It's just, I don't know why, but that's what happens. Yeah. Okay, going back to this question, I'll just do this, and then that might be it for today. But if you come across this kind of question, now generally you don't see questions like this, but there is no reason why you don't know how to do it, because whenever you have to explain the bonding in a compound, you have a periodic table available to you, and what you need to do is find the two elements involved. Anybody got anything? Physics forces required practice with the math and trolley. Yeah, I can just go through it briefly. The bonding in francium bromide. What you need to do is look at which group of the periodic table these things are in, which you have on the periodic table, remember. Now, I can tell you that that is in group 1 and that is in group 7, and that is all you need to know. Because if you know that an element is in group 7, group 1, you know it has one outer shell electron. If you know it has one outer shell electron, you know what it needs to do to get a full outer shell. Loses one. If it loses one, you also know what ion it forms. It forms a 1 plus ion. You see how we've got all of that just from knowing what group of the periodic table it's in. Similarly, in group 7, we know everything has 7 outer shell electrons. If something has 7, what does it need to do to get a full outer shell? Gains 1. If it gains 1, we know what ion it forms. It becomes a 1 minus ion. And then all you need to do is explain that because you've got positive and negative ions here, oppositely charged, the oppositely charged ions attract. And we now have francium bromide. I think that's it. Is that francium FR? Yeah, probably. And that's it. So my point is, just because it's something that's unfamiliar to you doesn't mean that you can't figure out what goes on. Right, last thing then. The required mass in the trolley is tricky, that one, because it varies a lot in terms of how it can be done. But I will explain. So in physics, there is a required practical to do with acceleration. And it looks a bit like this. So the idea is to find out whether the force on the object affects the acceleration on an object, which it does. The bigger the force, the bigger the acceleration, but we just need to prove it. We essentially, I'm not going to show you all of this, but we essentially set up a practical that looks like this. So we add weights to here and then let it go. And this toy car obviously flies across to the right. The tricky part of this is how you then measure the acceleration, because the whole point is you're trying to relate the weight here, which is obviously easy because you keep adding it yourself, to the acceleration of this toy car. Now, there are numerous ways to measure acceleration, but well, this mentions a formula, but I don't think you need to worry about that formula. Um, Yeah, fine. Well, I mean, the point is, you can do it using a computer, or you can do it using these light gates. There's a thing called light gates here, um, where it goes through one, it goes through the other, and a computer works out the acceleration. Or there is a formula, it's like v squared equals u squared plus 2as, which allows you to work out acceleration based on how far the thing traveled and how fast it was going. There's a few different ways to do it, so that's not the important part, though. The important part is understanding that what we're doing is adding weights to this and seeing whether that affects the acceleration of this car. And what will happen is that the more weights you add, the faster or the greater the acceleration is going to be. That is always what happens. That's one version of this practical. The other version of this practical is whether or not the mass of the car itself affects its acceleration. And again, it does, And the way you do that is you add weights to the car, not anywhere else, and then do the same experiment. You measure the acceleration and you compare it to the mass of the car. Again, the answer is going to be that the heavier the car is, the less it accelerates. That's always going to be the answer. But this is just a case of proving it using an experiment. The length of the string, I mean, I think it just needs to be long enough to be able to calculate um, to be able to measure a certain amount of distance. Do you see what I mean? Like, so that you can actually measure the acceleration over a certain period. But the length of the string, generally, no, it shouldn't affect it. I mean, it probably no. I would say no. I don't know the answer, but I would say the length of the string doesn't. But it does need to be long enough for you to get enough distance to be able to actually accurately measure the. So you don't want it to be like there. I don't know how you're going to do it from there. Um, so that's it really. Um, the length of the string, yes. Each time, yes, you shouldn't change the length of the string in that each time you do this experiment, the length of the string should probably stay the same. You shouldn't make it shorter and then longer and stuff. Similarly, if you're doing the experiment where you're adding weights here, a control variable will be the mass of the car. Assuming that's staying the same in that scenario. But as if you're changing the mass of the car, then this force should stay the same. That should be the control variable. That's why it's important to understand which version of the practical you're doing each time. Um, yeah, so if you're doing the one where you're changing the forces here, then the mass of the car should stay the same. If you're changing the mass of the car, then the force here should stay the same. So it's just understanding which version of the practical you are doing. Yeah, I think that hopefully covers it. But the reason it all is true is because there is a formula which is F equals ma. Essentially, if you that's the F is here, that's the force on the vehicle. If that gets bigger, the acceleration will get bigger. Whereas if the mass gets bigger, then the acceleration has to get smaller. So it's really just that formula explains it. As is the case with most physics required practicals, there is a formula associated with them, whether it's specific heat capacity or wave speed or resistance or density, right? There's a formula associated with basically every physics required practical, which kind of explains what you're about to do. Right, assuming there are no further questions today. Just as a reminder, you can always there is a WhatsApp number, you can message it beforehand that I can prepare suitably for any of these should I need it. You will be doing some chemistry next week. As I say, I think year 10 are going to be doing organic chemistry, I think. Crude oil, fractional distillation, stuff like that. Year 11, you'll be doing the chemistry required practicals, which are very exciting. And we'll